Hi, everybody. Thanks for joining today. This is Jason Key at Harvard Medical School, Associate Director of SB Grid. Hello, everybody. It's a real pleasure today to have Scheres on our webinar. So I think everyone is excited to hear about new features coming in Relion 3.0. Sjors leads a group at the MRC in the UK. He's the developer of Relion, which I think, as cryo-EM tools goes, is certainly one of the most popular and has probably sold more NVIDIA cards than any piece of software that I'm aware of. So, Sjors, are you there?
Yes. Can you hear me all right?
Sounds good. Go right ahead.
Cool. Yeah, thanks, Jason. Thanks for the invite to speak for your audience. So today I'd like to discuss what's new in the orange version of Relion 3.0. And the first thing that's new is that whereas until now I'd been mainly programming on Relion on my own with kind of a limited part of the code being developed by Shaun, a PhD student who developed helical reconstruction. Now I have two postdocs who are helping me a lot with developments inside Relion's core and everything around it. So, and I will present work from both of them today. So there's Jasenko, who's developed Bayesian polishing and the CDF refinement and the Beamtilt estimation I'll speak about today. And then Takanori Nakani, he's been involved with the implementation and testing of multi-body refinement, has done lots of tweaks throughout the software, also to helping make all our kind of body of software around it work with the new implementations from Jasenko, and has been extremely helpful in providing user support. And many of you will have seen all his answers on CCPM email list. And then Li Dong is a new PhD student in my group who's working on automation of class selection in first instance, and her work hasn't led yet to incorporation in Relion 3, but hopefully it will become part of future versions. So I will also not speak about today about work done by my collaborator in Sweden, Erik Lindahl, who's in Stockholm, and they did the GPU acceleration of Relion 2, and now as part of Relion 3, they've done similar unwrapping of the very intricate loops in Relion's inner core, such that you can use vectorization on CPU. So now Relion 3 can run on multi-core CPU clusters much faster than before, about 5 to 10 times faster, and thereby it becomes, I think, in cost efficiency is kind of similar to the professional type of double precision NVIDIA cards. Whereas I think single precision gaming type of cards are still a lot cheaper than running on the CPU. Okay, I thought I'd just go from the top to the bottom in the kind of job type window here on the left, starting and highlighting what is new. So one thing that is new is that Takanori has made a C++ kind of reversed engineered version of Motion Core 2. People on Twitter have been calling it Relion Core 2. On the GUI, it's called Relion's own implementation. So we did this originally to be able to take forward more metadata involved in subframe alignment onto Bayesian polishing. Although in the end, it doesn't seem to matter very much. So what distinguishes it from what it tries to replicate Motion Core 2 is that this is CPU only. It's multi-threaded, and provided you have a, say, 12 or a 16 core machine, and then you can use integers times of the number of frames that you have for the number of threads, such that each thread has an integer number of frames to process, then speeds are better or equal to than GPU processing in the UCSF version. As I said, the original argument of doing this was passing around of metadata. One difference as well is that this is open source and free for all. So there's, even if you're a company, you can use this. And overall, Relion is all open source, and we take a point in that open source software, even though people can copy pieces and put it into their own and so on, that you could perhaps perceive as a threat to your own software. Ultimately, that is impact that your developments make, and a field grows faster if people use open source, which other people can build on. And I will show you examples also today of things that we've taken from other open source packages and incorporated into Relion. So in the automated particle picking job type, one thing that is new, you can now provide a 3D reference, and this was upon popular request, and we use this in our fully automated on-the-fly processing scripts that I will mention later on, where you take a 3D map, you project it in with a given angular interval into all different orientations, and then you use those for reference-based picking. Another new thing is this Laplacian-of-Gaussian picker, and that's there's a second tab here, the Laplacian tab, and this is meant to be Relion's for the moment approach to do reference-free auto picking. It's based on a Laplacian-of-Gaussian filter, which is sensitive to blobs of a given size, and then you can vary this from a minimum to a maximum size. So if you have more or less spherical particles, these minimum and maximum size can be very similar or the same. If you have very elongated particles, then you can reflect this by very different values of the minimum and the maximum diameter. The filter itself is, we've tried to normalize the threshold such that just default values give reasonable picks, if not perfect picks by far, but enough to get you started. Again, we'll see this later on in the completely automated way of processing your data. We aim to get thresholds that are valid for a whole wide range of different types of data sets. Now, the way the program works internally, it will search the micrographs for sizes of blobs that are way smaller than you've specified from your minimum to your maximum diameter. It will do the sizes of the minimum, the average of the two, and the maximum size. That's your expected size range. And it will also search for blobs that are way bigger than your accepted particles. And the too small and the too large searches serve to identify regions of the micrograph where we think your particles are not there. So these are trying to find the true negative areas and focus your searches on areas where your particles are of the right size. This is a very favorable test case, perhaps just a ribosome data set on a direct electron detector, so contrast is very beautiful. You can see most of the ribosomes are picked. There's some false positives here, which result from a compromise that at some point there's just one general threshold to pick them all kind of different quality data sets. So the idea is that at least for a wide range of possible quality data sets, you get at least a subset of particles that is good enough to then start your on-the-fly processing approach, which I will discuss later. So I personally wouldn't rely on just log-picked particles for your final data processing, but it might be a good alternative to what used to be normal in Relion, that you first pick several micrographs or a few dozen of them by hand, and then use reference-free 2D class averaging to generate references. Now you can kind of start from log-picked micrographs and go all the way without any user having to pick particles by hand. Then on the 2D classification job type, there is a new option to use fast subsets, and this is particularly useful for large data sets. And this is an idea that we got from Nico Grigoriev's and Tim Grant's paper on system, which is published in eLive. And this is to use fewer particles in the early iterations, because if you have picked 1 million particles from your 2 days worth of data collection, then 2D classification will assign random orientations to all of the 1 million particles normally, and calculate spherical, like blobby references. And then during the first few iterations, you pass over each iteration 1 million particles, and these circular references become slightly less circular in the first iteration, and then slowly converge towards the hopefully representative views in your data set. But in practice, you don't need to pass over 1 million particles in those first few iterations, because the references are still very low resolution, so you can get away with much fewer particles. So we use some heuristics there. It's hard-coded inside the core. It's similar to what Nico does. And I personally probably wouldn't use it if your data set is not bigger than, say, 100,000 particles, then the differences aren't that big. But if you go beyond that, then the data processing will become a lot faster. And what we've seen, the results are, of course, different, but they're not qualitatively worse than just passing over all data. Then initial model generation in 3D. Already in Relion 2.1, we implemented stochastic gradient descent, an idea originally proposed by Ali Punjani and Marcus Brubaker in Toronto, and one of the bases of the CryoSpark package. So in 2.1, our implementation was suboptimal, I would say. So it would work if the data was very good, but CryoSpark definitely worked a lot better for many data sets than the Relion initial model generation. So in Relion 3, because CryoSpark 1 was still open source, so we could actually have a look around, and we actually implemented something much closer to the CryoSpark algorithm with most of the parameters. Those who've used CryoSpark will recognize them now. So you can do things very similar in Relion, and we've seen that actually has made the gap between CryoSpark and Relion initial model generation a lot smaller, if not absent in many cases. So initial 3D model generation within Relion de novo has become a lot more robust than it was in previous versions. You can now also refine multiple initial models at the same time, etc. So that's inspired by CryoSpark. Now, then one of the big changes in Relion 3 is the inclusion of multi-body refinement. And multi-body refinement, as I said, was co-developed by Takanori in my lab. And it is basically, it builds upon the concept of focused refinement with partial signal subtraction, which was already available in Relion 2. And the idea here is that you have a part of the protein complex which you are interested in to refine separately from the rest of the complex. For example, because it is flopping around, or because there's some heterogeneity, other types of heterogeneity happening there. Now, in schematics here is then a part we're interested in red, and the part we're not so interested in yellow. This is gamma-secretase, where we first did this back in 2015. The experimental images are, of course, a projection of both the red and the yellow part, and then there's noise on top. Now, already on ribosomes and so on, back in 2013 and 14, we would do focused refinements where we put masks on one subunit, and then at every iteration, in order to focus refinement on that subunit and be kind of insensitive to changes happening in the other subunit. The disadvantage of that is that the masked reference projection, for example, if the mask would entail only the red part, then the reference projection only have signal of the red part of the protein, but the actual experimental image has noise, but it has the red part and the yellow part of your complex. So the comparisons between the experimental image and reference projections from masked refinements alone are inconsistent. The yellow part is missing. So the idea there was to then subtract, to make a mask around the yellow part and subtract in silico the reference projections from the yellow part from the experimental image, thereby generating a new version of the experimental image which only has projected density, hopefully, if the subtraction were perfect, of the red part that we're interested in. And then the focus refinement thereby becomes the comparisons with the masked projections become consistent again. Now, that was then used by Kelly Nguyen here in Kiyoshi Nagai's lab on the spliceosome with great success. And I think many people have used focus classification, focus refinement to great success. Now, multibody refinement, it goes one step further, and it provides a iterative partial subtraction. So you don't just do this once at the beginning of a refinement, but you do this iteratively at every iteration you subtract. And you now have not just one part of the structure that you focus your refinement on, but you can divide your complex into as many rigid bodies as you would like to refine separately. And the multi-body refinement will do the subtraction during every iteration iterative. I'll come back to that for an example of a 3D floppy protein that's here. So imagine we have a three domain protein, and the M domain and the R domain and the C domain are connected by flexible linkers, such that you have independent motion of M with respect to R and of C with respect to R. The way multi-body refinement works is just like focus refinement on the M domain. We're going to make reference projections of the RC domain and subtract them from the experimental image, which has MR and C domains, which leaves an experimental image where the M domain is still present and the R and the C domain are subtracted. And then we do a refinement of this image with the reference projection of only the M domain. Now, that will give a new set of orientations for the M domain, which hopefully is better than the initial guess that we got from our consensus refinement, where we just imagined that this was one rigid structure and we refined all the images just by projection matching against it. Now, you can already see here the partial signal subtraction wasn't perfect, and that is because the orientations of the R and C domain perhaps were not perfect either. So the M domain had some problems in its orientation, but also the R and the C, and that depends on how the different domains flop around in this particular instance of the experimental particle. So we do this for the M domain in the first iteration, but simultaneously we also do this for the R domain. So we subtract the M and the C domain and do a focus refinement on the R domain. That gives also new orientations for the R domain, and likewise also for the C domain. In the next iteration, the subtraction of the RC signal for the focus refinement of the M and all the other ways as well becomes better because these angles are better. So you iteratively do better partial signal subtraction, and while you accumulate now 3 sets of orientations for each experimental particle, which express the relative orientations of the M, the R, and the C domain with respect to the overall consensus orientation from the first refinement. And this you iterate through all the way until the orientations don't change anymore, and the resolutions of the separate reconstructions for the M, the R, and the C domains don't improve any longer. So in that way, it's similar to just auto-refinement. So the example I'll show you is from another spliceosome complex, the B complex, which is also done in the Nagai group. And Takanori made masks around 4 different domains. So the core domain here in the middle was the highest resolution in the consensus structure. You can see quite nice structure. The foot domain was still okay as well. And then the helicase domain here already was getting a lot more fuzzy. You can see density disappear here. And for the SF3B domain on the top, the density is very poor, and this threshold would almost completely disappear. So there is partially dependent, partially independent rotations and translations of these 4 domains with respect to each other. Now, the consensus map, and that's here shown in the top panel, it shows different slices along the z-axis through the consensus map. So this is the core domain here, which is very nicely defined, and then the foot is down here, so I'm a bit lower in Z here. You can see the foot gets a bit fuzzy, but it's also still okay. And then the SF3B and the helicase domain, this is the helicase and this is SF3B, are a lot worse. So they are very, very fuzzy in the consensus structure. Now, after multi-body refinement, doing this with 4 bodies at the same time, you now get 4 separate reconstructions of the individual bodies, and the core improves a little bit, but it was already quite good. The foot domain, you see this was quite fuzzy here, this was a bit fuzzy. It gains in resolution, although already it was also quite good. And the most dramatic differences are seen for the helicase domain, especially here at the outside, and for the SF3B domain. So that's also then reflected if we do the local resolution estimation on the consensus structure, local resolutions beyond 10 angstroms here at the top. And if I now place the individual bodies of the 4-body refinement on top of each other, that's a problem. We don't really know what happens at the interfaces, but I'm just rigid body docking them into the consensus structure here. Then you can see that the local resolution is improved throughout the structure, and the same is recapitulated by the FSC curves before and after multi-body refinement. Now, one interesting aspect of multi-body refinement is that you get the set of orientations for each of the bodies for every experimental particle. So every experimental particle now has 4 sets of orientations. And you can do a principal component analysis on those orientations to find out what are the most common type of motions that happen in the entire set of particles. And then you can just look at how much variance each of those motions correspond to. And here the first motion has most of the variance. The second motion was the one that the Nagai group thought was most interesting. And you can then make movies along this type of motion to get insights on how the molecular complex, what kind of motions are present. And you can see for the spliceosome, that the SF3B and the helicase domain kind of have this concerted motion with each other on top of the core, whereas changes here in the foot are a lot smaller, which kind of which agrees with the resolution of the foot originally being quite good already. So I think this is one of the main attributes of the multi-body refinement compared to the partial signal subtraction and the focus refinement, is you do everything at once, and that does allow you to get overall motions out with these kind of movies. Now, another major improvement in Relion 3 compared to Relion previous versions of Relion is the way we do the per particle based motion correction, which traditionally was called particle polishing in Relion, and because it generates shiny particles where signal to noise ratio is improved. Now, Bayesian polishing, as its name, this was done by Jasenko. Bayesian polishing, as its name suggests, uses a regularized likelihood function to follow the per particle motion tracks through the movies of your direct electron detector. Now, the likelihood function is just defined by the movie frames, and the prior that you need for the regularized likelihood function, or the regularization terms, are based on smoothness in velocity, so you can't have very big accelerations or deaccelerations on the acceleration, and then also on the observation that neighboring particles tend to move in similar directions. So we impose this with a Gaussian prior on keeping the tracks of neighboring particles the same. And then Jasenko's algorithm looks at, for each particle, for each micrograph, it looks at all particles simultaneously and all movie frames simultaneously to find these most likely track given the regularization terms. And the output of that is then going to all these PDF log files where you can find these kind of plots, and we present in the paper on this, which will soon be published in International Union of Crystallography journal. We use plasmodium ribosome, beta-galactosidase, and gamma-secretase. The tracks alone are kind of interesting to look at, but they don't really tell you much what's going on. But one thing that's interesting is that if you look at the fall off of the signal with spatial frequency for each of the movie frames, and already in previous versions of Relion, we modeled this with the B factor, and B factors being more negative here means there is less high spatial frequency content in the images. Then the old polishing method for the Plasmodium ribosome captured the initial motion very poorly, which led to always the first few 2 or 3 frames having relatively poor B factors. And in the new polishing, these tracks initially are followed much more precisely, leading to much higher initial B factors for the first few frames. And that's where radiation damage is still the lowest. Those are your most important frames. So by rescuing these initial frames, you can get more out of your data. And that's, I guess, the main contribution of the Bayesian polishing. So that then leads to resolution improvements consistently. Oh, I'm running out of battery. Sorry about that. That then leads to resolution improvements for all the cases that we've run this on. And another way of looking at it is that you can look at these B factor plots where you plot the logarithm of the number of particles versus 1 over the resolution squared, and you can then see that the Bayesian polishing gives you better B factors, or in other words, you can get to the same resolution, and for these data sets it then turned out more or less for 60% fewer particles you can get the kind of same resolution. Now, the second development by Jasenko is this other job type, CTF refinement. And the CTF refinement actually includes 2 different types of calculations. One is a refinement of the CTF parameters. Basically, defocus per particle is the most important thing here. And the other development is estimation of how much beam tilt is present in your data. So the beam tilt leads to the coma. If you haven't done your coma-free alignment of the scope well, then you have beam tilt in your data, which leads to phase differences in the images, which you can estimate and correct for. So if you want to do per particle defocus estimation, then that includes one more parameter for each particle. So if you have 100,000 particles, that gives you 100,000 parameters. That's enough to overfit your data. So you have to do this using this gold standard separation of 2 halves. Otherwise, you will get overfitting of your data. This is just a technicality, but which is implemented in Relion, and you don't need to worry about it. But you would if you would like to do this in other pieces of software, perhaps. Now, the reason why we think we can do better than just CTF Find 4 or GCTF in the beginning, where you estimate defocus from power spectra of micrographs, is that the power spectrum, this is kind of the power spectrum signal. So you see the rings, but they lie on top of quite a bit of background noise. And here the dotted gray lines kind of indicate how much noise there is on this signal. Whereas if you do a per particle comparison with the reference projection of what the signal is in the particle images and compare that to the reference, then you can see both the positive and the negative modulations of the CTF. You can see the zero crossings, and importantly, the amount of noise on this type of signal compared to the amount of signal that there is much better. So you can estimate your defoci more accurately if you do reference-based, if you know what the signal is, basically. So we tested this, and this is not necessarily the recommended way of running this, but just to show you how far this can go, we tested this on the data set. I guess it's from Dimitri Liumkis on the Hemagglutinin, which was tilted at 40 degrees to overcome preferred orientation. So we just started from a per micrograph GCTF defocus estimation, which is pretty badly off for the parts that are much closer and much further away from focus due to the 40 degree tilt, of course. Now, originally, I think Dimitri published this data set at 4.2 angstroms resolution, and then the other Dimitri Tikhonov processed these data in warp and then used cryospark to get to a 3.2 angstrom resolution. So when we use Relion 3 for this, we actually had to use 4 cycles of CTF refinement, because the initial reconstruction from the average defoci of the entire micrographs only goes to about 7 angstroms resolution. And if you then refine defocus, you suddenly make a jump to close to 4 angstroms resolution. So that goes from the gray to the orange line. But once you have that reference, you now have a much more detailed reference. You can then again do better at defocus refinement. So that's why if we do this 4 times, then things start to stop, improve. And then we do the Bayesian polishing, we classify a bit and do another 3 rounds of defocus. Then in the end, we end up with a 3.1 angstrom structure. And the kind of features that we see in the map as we go along this process reflect the kind of improvements. And so does a FSC calculation against atomic model. If you then look at the refined defoci for particles within 1 micrograph, you can see beautifully this tilted aspect of the micrograph back in the defocus values, and there are probably a few outliers where refinement has gone off. But overall, you can rescue these defoci, which kind of illustrates that you can correct defoci values, which are here in quite a substantial amount of quite a substantial range of 5,000 angstroms. Now, Beamtilt estimation, Relion since version 1.4 or 2.0, I think, had the option to correct for Beamtilt, but we never knew how much Beamtilt there was present in our data. And it was Jasenko who developed a method to estimate the amount of Beamtilt from, again, comparisons of the individual particle images with clean reference projections and see if there's systematic deviations in the phase errors in between those images. Now, one thing that this allows us now to do is to collect images using beam shift rather than state shift. So what we typically do is we focus somewhere on the carbon, and then we take a picture in the middle of the hole, then we move the stage to the next hole and repeat. Now, after you move the stage, you have to wait for drift to settle, so this becomes a rate limiting step. So therefore, it's quite popular to use beam shift to collect 4 images in each hole and then move to the next hole. Now, if you use beam shift in the microscope to shift your beam, that actually introduces a bit of beam tilt. Now, the amount of beam tilt you get from shifting within a hole is not a problem, but what you can do, what would be potentially even faster, and this is together with Wim Hagen at the MBL in Heidelberg, is to take images in 9 holes, 4 images in each hole, for example, and then move the stage to a next position way in the middle of another 9 holes. So then, therefore, again, increasing speed of data acquisition because you don't need to shift the beam. And Wim was developing methods using the optics of the microscope to correct for the actual beam tilt that shifting the beam so far out induces. So you can use the microscope hardware to take images here that do not have beam tilt in them. But as a control, Wim also collected data where he just ignored beam tilt and just took the images using the beam shift anyway. So we thought that would be a nice data set to test our method of estimating and correcting for beam tilt on. So these are on Apo-ferritin. He took actually 5 images per hole and then used 9 holes. And there's one data set where he uses active beam tilt compensation. That's his hardware method to correct for beam tilt. And then his controlled data set where he didn't do this. Now, his active beam tilt compensation works very well, and you then get a 2.1 angstrom or so structure out of these 400 images of Apo-ferritin. Now, if you don't use any correction for the beam tilt, nor hardware nor software, then you get a much lower resolution, which is, I think is about 2.8 or 2.7 angstroms. Now, by estimating the amount of beam tilt from the particle images and then correcting in Relion for it, we get back to more or less the same reconstruction, perhaps even slightly better than what Wim got in his hardware correction. And this is just to show you, this is for the 9 holes. So this is for each stage position, you take an image in the central hole, which will have very little beam tilt, and therefore the systematic differences in phases between the Fourier components, that's what we're looking at here at negative blue and positive red and yellow, are more or less absent when you take images down to the central hole. But if you shift the beam upwards all the way to the next hole, then you introduce these phase difference in the direction perpendicular to that shift. So you get positive phases on one side of the spectrum and negative phases on the other side. And Jasenko fits functions. This goes to the cube with resolution. We know how they go. So Jasenko can fit these functions through them and thereby estimate very accurately what the amount of beam tilt that you get for each of these holes and the direction of the beam tilt too. And then the correction for that leads to these huge improvements in resolution. Now, one other thing that's also new in Relion 3 is Ewald sphere correction. And this, at the moment, at 300 kV, only matters for really large particles, probably mostly icosahedral viruses. It will become more important, and Richard Henderson and Chris Russo are now trying to push for lower resolution 100 kV microscopes, perhaps with a FEG. Then these things will become more important. They also become more important with higher defocus. But if you do have a large virus, and this is an example that we show, P22 virus, which is about 700 angstroms across, if you do use Ewald sphere correction, according to the algorithm that was proposed by Chris Russo and Richard Henderson earlier this year, then you can actually, from not doing any Ewald sphere correction, you can improve on the resolution using Ewald sphere correction. I have to say this is not implemented inside Relion Refine. This is only part of Relion Reconstruct. So after you've done your refinement, you need to take the data.star file and do reconstruction for each half of the data using Ewald sphere correction. Because you don't really know which is the correct hand, you have to do this for both one. You could have flipped your data around an odd number of times during image processing. So in practice, you need to try both hands in the Ewald sphere correction, and one will become better, the green line, and one will become worse, which is the orange line. Then I mentioned in the beginning that we've now worked on scripts to do on-the-fly processing without any user interaction. We call this relionit.py because you just need to rely on it. There's no more space for you to interfere with your data processing. And this is just a screenshot. It's a script written in Python, and it will schedule and execute jobs just within the normal Relion GUI pipeline. So it's if you like, it's a Python. Well, it's basically a command line interface into that what you normally would deal with in the GUI, and that allows you to write scripts around it. And then we used Python, but you could have used a Bash script as well. And we hope that these Python scripts, we provide two, one for on-the-fly processing called relionit.py, and one to make B-factor plots, which I showed before, which is the logarithm of the number of particles versus one of the resolution squared after refinements. So we hope that these scripts will be an inspiration for others to write their own kind of semi-automated procedures. So the relionit.py script will iteratively import micrograph movies. It will do the motion correction, CTF estimation, do this log picker, and then go from extracted particles straight into 3D SGD, from there on into 3D classification, use the best class to do reference-based auto-picking, and then get a final kind of set of particles, which you can then use for 3D classification or refinement. And to test this, we used gamma-secretase, which was originally collected here by Xiaochen Bai, postdoc in my group, who's now at UT Southwestern. And we chose this data set because this is not your kind of bulk standard test data set like Apo-ferritin or beta-galactosidase. This is a 130 kilodaltons ordered mass membrane protein complex. So this is definitely not very easy data. And we then compared how Xiaochen's very expert selection of particles, which gives a 3.2 angstrom map in Relion 3. It gave a 3.4 angstrom map in Relion 1.3 by Xiaochen. But so the improvement of gray to green is due to Bayesian polishing and CTF refinement, etc. But this involved Xiaochen looking at 6 subsets of the data, do individual 2D classification, select all the nice classes, etc. How that would compare with a fully automated processing by the script is then given by the orange line. So that gives a 3.3 angstrom structure. So fully automated processing of gamma-secretase in Relion 3 is slightly better than Relion 1.3, but it's not as good as Xiaochen would have done in Relion 3. So, but the difference is getting smaller, and we will keep working on this script. This is definitely a work in progress, and I think not too far in the future you'll see that all the way up to kind of consensus refinement will probably become relatively easy to do in a fully automated manner for many data sets. I just want to finish with 2 data sets just to show what Relion 3 can improve compared to what you can achieve with it. One is this Empire entry by Sriram Subramanian's group on beta-galactosidase, where they initially published a 2.2 angstrom map, and then a map that they said was atomic resolution. So the Bayesian polishing gives you these kind of B factors. So again, whereas previously Bayesian polishing would start at -200 here or so, now the first few frames have a lot more high resolution information contents after dealing well with the initial motion of the particles, which is very fast. Surprisingly to us, this data has a significant amount of beamtilt. There's about 0.2 millirads in the x direction, and you can see that here in this phase pixel phase difference plot that Jasenko's program writes out. So there's systematic negative differences on one side and positive differences on the other side, which is a clear sign of beamtilt. So this is one of the reasons why we could improve on resolution. Sriram used per particle astigmatism in his atomic resolution paper, and we think that might have been partially describing the effects of beamtilt there. Now, this is how our processing scheme starts. We start with a large set of auto-picked particles. We do defocus refinement and tilt refinement. If you do just one of them, either defocus refinement or tilt refinement, you go from the gray FSE curve to the dotted or the solid line orange one. If you do both of them at the same time, their effects are cumulative. You go to the pink line here. Then the effect of Bayesian polishing on this data set is quite large as well. You go to the purple line. Then we tried a little bit of classification and do another defocus and tilt refinement. Improvements were a bit smaller. And you can see throughout this process that the map actually gets better and features start to appear that should be there. So this final green FSC curve corresponds to a 1.9 angstrom resolution map, where we see beautiful rings, beautiful holes in 6-membered rings, but not yet that much in 5-membered rings, which is as expected at 1.9 angstroms resolution. I told you we have this Python script to calculate this B factor plot, which makes automatically for you subsets of the data set and does refinements for each of the subsets, and then it looks at the post-process 1 over resolution square, and it makes this nice PDF plot for you. And from that, we estimate now that the B factor for this data set is 56 squared angstroms. And that compares with 91 squared angstroms in Relion 2. So you knock off more than 30 squared angstroms from the B factor going from Relion 2 to Relion 3 due to a combined effect of the focus refinement, beamtilt correction, and Bayesian polishing. And knocking 30 squared angstroms off in Relion 2 then improves resolution from 2.2 in Relion 2 to 1.9 in Relion 3. And that's on this plot. So the orange version of Relion gives the orange line, and the green version, Relion 2, gives the green FSC curve. This is the FSC of Sriram's model versus our map. So we didn't do any attempt at re-refining Sriram's model. We just took that at face value, and we see if we the reported resolution of 2.2 to 1.9 is reflected in an improvement in the FSC of model versus map, which is indeed the case. Interestingly, Sriram's map, which he claimed was at atomic resolution, is correlates with his own model up to the gray curve. So the Relion 3 map is better than the atomic resolution map from the recent structure paper. Although I think calling it atomic resolution is probably not correct yet. So Apo-ferritin is then the last case I thought I'd mention. This is another data set by Wim Hagen, collected at the EMBL, taking now 7 images per hole, not doing the beam shift all the way to other holes. So this has just conventional stage shift. I think it's a bit older data set. So he collected 1,200 movies at 0.8 angstroms per pixel, just to see. He thought he'd see how well his cryos would do. We pre-processed these data using the Relion.py script to get the initial set of particles. We then did 2D classification, selected nice classes, which we did CTF refinement and Bayesian polishing on. There was hardly, we tried Beamtilt estimation, but there wasn't hardly any Beamtilt in this data set because I think Wim aligns his microscope very well. And that then led to a reconstruction to 1.65 angstroms resolution. So this is very close to 2 times this number. I think we're only 2 or 3 shells away from the Nyquist frequency. I do think this resolution estimate is true, though. We see beautiful rings in prolines. We can see even bigger blobs for sulfurs here, and overall, I think density looks pretty good, reflective, as you would expect for a 5 angstrom resolution map. That was all I had to say. You can download Relion 3. As long as it's beta, we have this download on Bitbucket, and then we'll hopefully soon go stable, and then we'll move everything over to the GitHub as well. So thank you for your attention. These are all the people involved. Jake, and I mentioned Eric and Takanori and Jasenko and Liji. Eric, Björn, and Daria are the people in Sweden, and Jake and Toby maintain our computing infrastructure. I am very grateful for all users who provide us with very insightful and detailed bug reports that just don't say it doesn't work, but actually tell us what goes wrong. And I thank you very much for your attention.
That was great, thanks. There's just been a couple of questions in the chat, if I can pass those over to you.
Yes?
So we have one from Longfei, which is how to use multi-body refinement on proteins with high symmetry.
That's a good question. If you have high symmetry and you want to maintain that symmetry, you're imposing that all the different bodies, they will flop. They have some motion with respect to each other. Otherwise, you wouldn't do multi-body refinement. So if you want to still refine multi-body in a high point group symmetry, then your basic assumption is that all the motions still obey that point group symmetry, which may very well not be true. So if you have a kind of typical example of an icosahedral virus from which floppy domains are sticking out of, then the floppiness of those domains probably do not obey icosahedral symmetry. And then rather than using multi-body refinement, you should use something a program called Relion Particle Symmetry Expand. And this procedure of symmetry expansion is described in a 2016 review in Methods in Enzymology, the book that was edited by Tony Crowther, which is all about classification of images in Relion. And that describes on how you can expand your data set, which would have 60-fold, which would become 60 times larger in the case of 60-fold icosahedral symmetry. You can then use C1 refinements on these expanded data to deal with that kind of floppiness. So in short, probably you wouldn't use multi-body on very high symmetry groups.
Okay.
Just taking off from that last question, I'm trying something similar with a virus, and I wanted to know how I could use particle subtraction on a symmetry expanded stack of refined virus particles, or should I just go back to using or refining the subtracted particles in C1?
Yeah, so you can use symmetry expansion first. So you'll get a much larger star file where each particle is replicated, and then you do subtraction using that expanded star file. So now each original particle will actually become multiple images on disk, each where a different part of the virus is still there, the signal, and then you can refine all that in C1 or classify it in C1. There is also a new option on the particle subtraction job type, which allows you to provide the center of rotation from which you would like to do the particle subtraction. And that might allow you to then use smaller box sizes on your subtracted particles. Although that will require a little bit of scripting around some of the command line programs. That's not possibly only through the GUI.
Okay, another related question on symmetry is, what tools would be useful in Relion to deal with protein assembly that has a highly symmetrical region while other parts are asymmetric?
There is this localized masking approach that Shaun also wrote, and it's you can find it on the GUI on the wiki page of Relion on the left side. There's something just under the helical reconstruction. There's something on localized masking where you can kind of impose local symmetry during refinement in Relion. In practice, we have not encountered many cases where that actually yields a big improvement. Perhaps that's why we never published it. So in practice, we often just refine in C1, and you can then still average over the symmetrical copies afterwards to make them better. But inside the iterative refinement, it doesn't really help often. But it might be that your case is the first.
Okay, there's just a couple more questions, and then I think we'll let you go. So, somebody called SPL would like to know how well ordered does the multi-domain protein need to be for multi-domain refinement to work?
Oh, it doesn't necessarily need to be very ordered. As you could see, the spliceosome parts of it weren't very ordered at all. I think the main limitation in application of multi-body refinement is the minimum size of the individual bodies. Because you're going to subtract all of the rest away, there needs to be at least, I would say, about 150 kilodaltons worth of mass within each body. If that's the case, then quite big motions can be refined. Perhaps if you have a completely floppy bead of, a string of 5 beads and there's very large amounts of motion between each bead such that the whole consensus refinement would utterly fail, then that would become a limit too. But rather than that, in most cases, it is the minimum size of the individual bodies, which is much more limited than their amount of motion. So if you get a reasonable consensus refinement, and the individual bodies are large enough, then multi-body refinement could work.
So I guess thank you very much for the great seminar. That was really useful. It's certainly something we'll be trying a lot around here. I don't suppose anyone else in the room has any questions? No? Okay. So thank you very much.
Okay, thank you. Goodbye then.
Goodbye.